You're listening to the Good Question Podcast with Richard Jacobs. Our goal is to make each of our guests exclaim, that's a good question. I don't know the answer. Because when that happens, it means you, the listener, may be inspired to learn more beyond the interview and to ask great questions yourself that lead to new insights. In this podcast, we cover historical and current anthropology, comparative religion, and history. Welcome, and let's get started.
Hello, this is Richard Jacobs with the Good Question Podcast. My guest today is Lyle Small. He's the founder and interim chief technology officer of a topic called dye-drug conjugates, DDCs. They developed a proprietary enhanced chemotherapy delivery platform known as dye-drug conjugates. It has a tumor-targeting dye, other aspects of it that I'll let him describe so I don't screw them up. But regardless, welcome, Lyle. Thanks for coming.
Thank you, Rich. Appreciate it.
Yeah. Well, if you would tell me just a little bit about your background and then how you got to work with these DDCs and with chemotherapy.
Yeah. So I'm a nerd, I guess, is where I should begin. I got an engineering degree from Cornell in 1993 and didn't want to get a real job. And I love the idea of entrepreneurship. So I started a company and filed a couple of patents on thermochromic ink. So basically, this company, Chromatic Technologies, makes inks and coatings that are interactive. So things that change color with temperature or with light, pressure, or water, things like that. So we want to tell consumers something about their product that matters to them. One of our most famous applications are the mountains on the Coors Light can. So my claim to fame is that I invented the ink that goes on those cans. Now, I didn't invent the version that's on there now. We've had some substantial improvements since my version in 2006 or whatever, but that was something that I did. So
when you say thermochromic, like I guess you put your hand in the beer can and the color of the ink changes because of the temperature of your hand.
Well, actually, the temperature that it's set at, so we can set the temperature wherever we want. The blue mountains of the Coors Light can actually tell you when the beer is cold enough to drink. So, yeah, we've got this on 20 plus brands around the world, beer brands, and we've added about $450 million in annualized revenue to the Coors Light brand because of our technologies, and that's what Coors Light tells us. So, consumers, especially of Coors Light, care about having a cold Coors Light. Warm beer, at least here in America, is not something that's very palatable for our taste. So having an indicator that will tell consumers when it's cold enough to drink has been a big idea. And they advertised this like crazy back in 2009, 10, 11 when they launched in the United States.
Well, I don't mean to go off topic, but this sounds like something that'll be very useful. Be a lot of useful for a lot of foods, by the way, to see if they're safe to consume, or maybe you have an ink that once it transitions in color, it can transition back, which would indicate that the contents have been gotten up to the wrong temperature and are now spoiled. I mean, I would guess all these things have been made subsequently.
Yeah. We have thermochromic inks that will tell you if a vaccine has spoiled. Whether a roofing membrane has been sealed appropriately, whether a high-pressure processing food has reached the appropriate pressure. So indicators of all kinds, all related to dyes that are microencapsulated. We've been working on this for 30 years now, and as of January, I've turned over the role of CEO to a much more qualified gentleman who is taking Chromatic Technologies and running with it. It's been a pretty successful business over the last 30 years, but I think its best days are ahead of it, especially now that we've got a new senior team involved. It's doing great. Now I've got a new project that has to do with cancer.
Let's get into your current work now with these these dye-free conjugates. What are they?
Yeah, so back a long time ago, we're a dye company, as I just mentioned, and I'm looking for dye chemists all the time because we're trying to find new dyes inside of our little microcapsules and having funky things.
You should tell people that you have a very colorful past of all the dyes and stuff you worked.
Colorful past. Yes, I definitely have been in the color business a long time. So dyes are a central part this business that I started out of college. And because of that, because I'm hiring dye consultants, I come across a lot of different types of dye chemists. One of them was a gentleman who was close to retirement, but he had a really impressive background in dye synthesis. Worked for Kodak for years, had 80 patents to his name, this really impressive guy. And he had worked with a group of scientists at Georgia State University and Emory University and developed a dye that sticks to cancer cells. It travels throughout the body, sticks to cancer cells selected, and it'll stick to 30 different types of cancer cells. So one of the first things this gentleman did on his first day of work in late 2009, early 2010 is show me some pictures of some mice with glowing tumors, because this dye is fluoresces in the near infrared. So when you inject it into the mouse, you can put a near infrared lamp on the mouse and you can see the where the dye is, and it'll fluoresce in an orange color. It's represented as an orange color in a near infrared image. And so he had these cool images of mice, and he said, we're working on this to tell surgeons where to cut. Really cool stuff. And of course, I thought, well, that has nothing to do with us, so good luck to you. But I really need to focus on leucodyes for thermochromics and photochromics. He said, yep, that's great. Couple days later, I was watching a TV show about nanoparticles as a possible hyperthermia. In other words, a way to kill cancer cells using heat. So you would, if you were able to get the nanoparticles in a tumor, for instance, you could put the patient in a radio wave machine and heat up the gold. And this was a 60-minute show that was literally 48 hours after I'd just seen these images of these glowing tumors. And I thought, well, could we use the dye targeting agent for the nanoparticles? Because that was the big missing link for this technology that they were talking about on STEAM Minutes. And you can look it up on YouTube. It's they've got a couple of shows about it. And we decided we would try to stick this dye to gold, filed some patents, and then proceeded to make junk for the next 3 or 4 years. And it was just one failure after another. This career that I've chosen is basically to invent things. We make new things. We've got 40, 50 different types of products at CTI that you won't find anymore. I'm really proud of that, but it's a hard way to make it. Failure is a lot more part of my day than success.
But I could see, I guess already one application. If you're able to get dye to stick to tumor cells, you want to do radiotherapy, you could have it much more concentrated and directed. Yeah. Maybe even almost down to the individual cellular level, and just lays, specific ones to knock them out.
Right. Oh, yeah. The opportunities afforded by a tumor targeting agent are myriad, right? You had lots of different possibilities. If you can find something stick to cancer, specific antibodies stick to cancer. And that's actually an adjacent technology to ours. And antibody drug conjugates are, a conjugate is just two different molecules that have been hooked together. That's the definition of a conjugate. Antibody drug conjugates are just an antibody attached to a drug. And those types of molecules, specific antibodies, will stick to a cancer. And so there are they're one of the holy grails of cancer treatment, figuring out how to get chemotherapy where you want it to be and to not be where you don't want it, because these are just poisons, right?
Has anyone been able to dye exosomes or extracellular vesicles or any of the components in them so that when a cell creates them, it either puts them out or takes them in, that you can see that and see a gradient of the color as they, they're move?
So the answer is, I don't know. Maybe. I think it's a great idea. But this particular dye does not do that. There's a dye called indocyanine green, ICG, that is approved by the FDA for staining cancer cells. So these two classes of dyes have this characteristic of sticking to cancer cells. There are other ways to tag a cancer cell, and I don't know all of the ways. I probably can't speak very intelligently about the other ways that people identify individual cancer cells or cancer cell types. I really just know about antibodies and about two particular dyes. But as far as exosomes go, maybe.
Okay. Yeah, just something to think about,
Yeah, I like it.
Okay. Well, yeah, keep going. So you describe what a conjugate is, an association of two molecules that are bound. So what are these specific conjugates you're working on for chemo?
Yeah, so we spent, 9 years trying to get this conjugate of nanoparticle dye to work. And we got the dyes we attached the dyes to the gold nanoparticles and tested it in mice. And we were able to get the dye to continue to function as a tumor targeting agent, which is a really tough trick. Very proud of that. The problem was that it was sticking to the liver as well. So we had to fix the liver problem, because you can't have a hyperthermia solution that heats up the liver and your tumor. That would be problematic for the patient.
Right.
So we spent 3 years figuring that out. And then we realized, probably way too late, that in terms of an investable technology, using nanoparticle was just never going to fly. Getting the device big enough to get an entire human in front of this radio wave machine to heat up gold all over your body was never going to happen. We couldn't find somebody that would build the device, and even if they did, it would be too expensive and take too much juice in a hospital. Somebody told an electrical engineer said, "Yeah, you'd probably dim the lights in the hospital every time you turn this thing on." So
yes.
It turned out to be another blind alley. And about that time, been thinking about other ways that we could use these dyes as targeting agents. And obviously, chemotherapy was on the table early. Back in 2016, we had discussed it. We shelved it while we focused on gold. Then 2018 and 2019, we really started to focus in on this, recognizing the problems with gold, and switched to chemo drugs. But in that case, you have to have a linker that will let the drug go. Once the drug is delivered to the site of the tumor, you got to turn it on. So the essence of our technology, tumor-targeting dye attached to a drug via what's called a self-immolating linker, means it dissolves, it cleaves itself and separates from these two things that it's attached to. So the essence... Yeah, this is back in 2019, filed some patents on that in 2019, 2021, and 2023. And then we started testing this combination, and started to see some results.
Oh, so, all right. These typical chemo drugs that, usually they act systemically. I haven't heard of ones that act locally. So what ones are you attaching to?
Yeah, no, these chemo drugs do act systemically. So that chemotherapy is medieval in the way it attacks tumor cells. This has been studied quite a bit. Chemotherapy actually is one of the most inefficient ways to deal with a person's tumor. But in most cases, it's the only option. Less than 5% of any chemotherapy that's administered ever ends up in a person's tumor. Usually, it's less than 1%, with an average of between 0.5% and 1% of any chemo drug that is injected into a patient getting chemotherapy will end up in their tumor. The other 99% of a chemo drug ends up in healthy tissues. It ends up in your gut, it ends up in your liver, it ends up in your brain. The cure, in this case, the treatment, is often worse than the disease. The number of people that die from chemotherapy is a shockingly high number. And, people have heard about the horrors of chemotherapy. Some people that have been through chemotherapy once or twice will forego chemotherapy rather than and face certain death. They don't have another round of chemotherapy. They know that they will die, and they still choose to forego chemotherapy because it is so painful, because it is so hard on the human body. So yes, chemotherapy acts systemically, and what we're trying to do through our solution is make chemotherapy safer and more effective. So we want to turn the drug off by attaching it to this system of this dye and cleavable linker, which we have done, and we've proven that the drug is non-toxic when it's in this form, and then let the dye travel throughout the system of the patient and stick only to cancer cells. And then once inside the cancer cell, the unique chemistry of tumor cells, special enzymes, and the acidic environment of a tumor causes this linker to cleave, which releases the chemo drug, makes it toxic again, and allows the chemo to do its job where it's intended to act.
So are you using inactivated forms of chemo that only get activated through this mechanism, so they're not the traditional chemo drugs?
Yeah, well, they're traditional chemo drugs, once they are released from the linker, they're the exact same molecule that's injected into patients.
When they're first given to patients, so the linker is attached, so therefore are they essentially an inactivated form that only gets activated when it's in the right spot.
That's right. And I want to be clear, we are not in humans yet. So we've only done mouse studies to this point. This is all preclinical data and small animal data.
What about, so if you have liquid tumors, I understand. Solid tumors, though, you have like hypoxic, anoxic, you have all kinds of different zones. So does it attack them from the outside and slowly eat them smaller and smaller until it gets to the inner material? Or is there anything that allows you to burrow through internally and start attacking?
You've exceeded my knowledge already. We don't know as much about the mechanism of how it attacks a solid tumor as I would like. What we do know is that we have penetration of this dye conjugate throughout the entire tumor itself. So we know that it's taken up. We don't know the rate of cell death in each layer of a tumor. So recognize that there's hypoxia going on in the middle, but we do, because we can measure the near-infrared signal of the conjugate, we can follow it, and we see it well absorbed by solid tumors. And the mechanism of action, that's what the FDA always wants to know. How does this thing function within the human body? What's the biochemical process?
In a mouse, have you had this attack a solid tumor? Well, not attack, just paint a solid tumor and then excise the tumor through surgery and then look at it, cut through it and see where the dye penetrated?
Yeah. I got some pretty cool pictures of some glowing tumors that have been excised from mice.
So what does that look like when you chop it through the center of it? Look, how far does it go into the tumor?
The entire depth of the tumor, throughout the tumor.
That's fantastic.
Yeah, it's amazing, actually. And one of the interesting things about the dye is that it's taken up by all cells of the body, but it's taken up at a faster rate in tumors and it's let go at a slower rate. So the entire process, the metabolic process that allows these dyes to accumulate in tumors, is really around how quickly things are taken in and expelled from cells in the body. So
do you have to wait for a while then? Like, so someone takes the dye, all the cells are affected, you wait till, what is it, excreted from most tissues, and then you go in with the chemo?
No, the chemo is attached to the dye. So the dye and the chemo drug are part of the same molecule. So what happens is, in the first 24 hours, we take a picture of our mouse that's had an injection of this conjugate, and the entire mouse glows. It just lit up like a light bulb. You can see every outline, the ears and the tail and the snout. Every part of the mouse is lit up with near-infrared fluorescence. Then on day 4, you can see there's still some glowing areas, but these mice are tumor-bearing mice. And you can see a very distinct dot on the lower flank of the mouse, only where the tumor is, that's beginning to glow very brightly, and the rest of it is fading. By day 9, the only thing that really glows is the tumor itself. And the beauty of this is that it doesn't matter that the conjugate enters healthy tissues, because once it's inside a healthy tissue, it doesn't let the chemo drug go. And we know this because we've tested it in healthy animals, and no observable toxicity.
Why is that? Why doesn't it let go in the healthy cells? If you could say it was not proprietary.
No, it's the dye's behavior has been studied a lot. So this dye has been around since 2006, and there's several labs that have looked at the pure dye and studied the mechanisms that cause it to do what it does. And so there's this channel in the cell wall called OATP. This is in the literature, and the dye travels through this channel, and it's absorbed more quickly in a cancer cell. There's more OATP channels, and because of the difference in the cell surface of a cancer cell versus a healthy cell, it's absorbed more quickly. Then once inside, it binds itself to the mitochondria of cells, and then it wants to stay there, and it stays much longer than in healthy tissues. And as this cycle continues, the healthy cells continue to eject the dye drug conjugate, and it ends up accumulating in tumor cells. We see near-infrared signal gets stronger and stronger in the tumor and weaker in healthy tissues. But because the drug is turned off and can't turn on until it's inside of a tumor cell, it's so far, all the indications are that it's safe for healthy tissues, because we've put a lot of conjugate into mice and seen very low toxicity.
This sounds ideal after someone gets surgery, if it's administered immediately with the surgery, because then if they miss anything, this would, get rid of it and kick its ass in the days right after.
Exactly.
I think this would be excellent to make sure that surgery is more efficacious, and maybe you don't even need long-term chemo with this.
Yeah, absolutely. And one of the most pernicious things about cancer is drug resistance. These cancer cells, the first thing that happens when you start attacking them with one chemo drug is that the cells start to become resistant to the drug. And pretty soon, the chemo drug is not effective at all. There's a cocktail of chemo drugs called CHOP. I think there are 4 different drugs, and I think the CHOP is the first letter of each of the drugs. The CHOP cocktail was created. It's one of the most effective chemotherapy regimens around today. It was created because of drug resistance. But each one of those CHOP chemo drugs is a poison. And, you go through CHOP and you will feel CHOP by the end. You feel like you've been drugged through a knothole, as my dad used to say. It is a very, very difficult experience. But you're much more likely to have your tumor shrink because the tumor can't become resistant. You're attacking it you're using different poisons to attack the tumor, and it's much more effective. Because we're using an approach that will lower toxicity, we believe, dramatically, the options for administering different types of chemotherapy via these dye-drug conjugates goes way up. If the patient isn't about to die because of the amount of chemo that's being administered to them, now you can administer 2 or 3 different chemotherapy drugs as long as they're attached to the dye, and you can start attacking the tumor specifically and from different chemical pathways. As different chemo drugs act differently, and they can prevent the cells from developing resistance, which is one of the biggest problems with chemotherapy.
Yeah, that's really, really cool. So how long until you were able to test it in organoids or in people?
Well, we've tested it in organoids, and it has performed very well. People is probably 2 years out. The process, we need to make the regulatory environment, which I am just now becoming aware of, is morass. It's very difficult. So an investigational new drug an application needs to be submitted. So we're probably 2 to 3 years out from beginning human trials. And the first stage is all about safety, takes a year or 2. The next stage is efficacy. That's phase 2. And then phase 3 is really about what is the right dose? How do you get this what's the practical way to get this drug into the marketplace? So we're probably a few years out from starting human trials, and probably a total of 7 years from now before this is widely available. But I think as I sit here today, based on all the data that I've seen, that day is going to come. We are shrinking tumors in mice in both lung and colon models, and the toxicity levels are very low. So even if all we do is reduce toxicity and make this safer, it's a huge win for patients all over the world. I think that this technology has the promise of changing I know that it's a big statement, has the promise of changing the way we treat cancer patients, at least with chemotherapy. So very exciting, the initial results that we've achieved. And we think that we've got so many options for attaching new drugs to this dye, different linkers, making it more and more effective while making it safer and safer, that really the upside is exciting. I think that we can get to a place where we will be more effective and safer than standard chemotherapy. And that's kind of a crazy thing to think, but
that's great.
It's a real possibility, yeah.
Have the mice have they gone totally into remission? Did tumors disappear, or they only shrink and stay?
No, they're not going to nothing. So we are very similar to current chemotherapy. One of the we used it as a control in our experiment, meaning the standard that we compare our stuff to is one of the most common chemotherapy drugs on the market today. It is well known, it is reliable, it's one of the CHOP chemo drugs, and we have used that as our control, and we are comparable to that drug. And for the first time out of the gate with 2 different conjugates and have both of them show efficacy, meaning they're obviously slowing or shrinking the growth of tumors, similar to a standard of care chemo drug that sells $1 billion every year with little or no side effects that we've seen so far, is a pretty incredible achievement. I'm very, very proud of our folks for what they've been able to do here.
It is amazing. I know you can't do everything, but what about, again, a protocol where you'll have a large mass, you'll excise that through surgery and administer this at the same time?
Oh, yeah. When you take debilitating toxicity off the table, all kinds of things are possible. But somebody comes out of surgery and you start administering poison that's going to bring them to the edge of death, there's a high likelihood that you will kill them. Chemotherapy is, the original chemotherapy drugs were cousins of mustard gas from World War I. To call them drugs is almost a misnomer. These are just absolutely the worst things that you can possibly imagine giving a human being, but they killed cancer cells. They also killed many of your other cells, but at least they killed your cancer cells. It is better now, of course. We've made lots of strides, but if you look at the data for the side effects from chemotherapy today, they're not a lot better than they were 30 years ago. But you still have to give a dose to somebody that brings them to the edge of death to try to save their life with chemotherapy, with modern chemotherapy today.
Yeah, that's crazy. Well, how can people keep tabs on your work? It sounds incredibly interesting. Where can they go to find out more?
So the company name is Lahjavida. It means the gift of life in Finnish and Portuguese, 2 different languages, LAHJAVIDA.com. So we're kind of under construction right now, but you can get at least a feel for what we're doing. Also, I'm on LinkedIn. I've posted several videos there giving high-level overviews of how the technology works and some of our data. So LinkedIn and the website is great, and also happy to respond to folks via email. And, we're going to continue to advertise this. We're going to have a presentation on October 17th at the Olympic Museum here in Colorado Springs, where we'll be talking about the results from this ongoing lung study that we're doing. We're trying to raise some money. This is not a solicitation, of course, but as my attorney continually reminds me, but we're trying to raise $5.2 million to get us to human trials. So we're going to be reaching out to folks that are like-minded and want to take a flyer on something that seems to be really cool right now.
Yeah, excellent. Well, very good, Lyle. Thank you so much for coming on the podcast. I appreciate it.
It's been my pleasure. Thank you.
You're welcome.
Thank you for listening to the Good Question Podcast. Please email support@thegoodquestionpodcast.com if you have any referrals to great guests for us to interview. Visit thegoodquestionpodcast.com to hear more interviews. And please help us spread the word by rating and reviewing us on Apple Podcasts, iTunes, Spotify, YouTube, or wherever you listen to this podcast.